You can go ahead.
Are we sure DNA is only a double helix? In April of 2018, antibody therapeutics researcher Daniel Christ from the Garvan Institute of Medical Research in Australia and genomicist Marcel Dinger identified the existence of a brand new DNA structure in living cells. The DNA component identified was the intercalated motif, or the I motif structure. It was first discovered in the 1990s, but only in vitro cells. Christ and his team have recently confirmed its ability to exist naturally in human cells, and not only synthetically created. An antibody fragment specifically recognizes and binds I motifs, highlighting their location in the cell with an immunofluorescent shine. Unlike the double helix, like pyrimidines on the same strand of DNA bind to each other. For example, cytosine and cytosine versus cytosine and guanine in the double helix structure. Erwin Chargaff's discovery of the ratio of bases in DNA has enabled Christ and his team to determine the new balance within the I motif structure.
So Frederick Miescher. Miescher discovered nuclein and DNA in 1874. His discovery laid the very groundwork for the DNA research, experiments, and molecular discoveries that followed. Without his findings on DNA in the nucleus of cells, the world would be very different. There would be no DNA research in general at all today. His breakthrough not only introduced DNA to the world, but inspired others to delve into the research world of deoxyribonucleic acid, our DNA. The very basic foundation of Christ's research is based off of Miescher's early understandings of DNA.
Just like Miescher, scientists like James Watson and Francis Crick are responsible for new DNA discoveries. Watson and Crick discovered the double helix structure of DNA. The discovery of the double helix marked a milestone in the history of science and gave rise to modern molecular biology, which is largely concerned with understanding how genes control the chemical processes within cells. Their discovery yielded groundbreaking insights into the genetic code in protein synthesis and paved the way for scientists like Christ.
Kary Mullis, the finder of PCR or polymerase chain reaction. PCR is often considered as one of the most important scientific advances in the field of molecular biology. With this revolutionary technology, it's possible to generate billions of DNA copies from a single strand of DNA. As a result of this, PCR is considered to be one of the most usable techniques used in medical and biochemical research laboratories. PCR is a huge technology advance in the study of DNA.